Hi, thank you. Actually, I'm just going to have a couple of asides about projects in my group. One about aphids, which is the boring one, and one about Littorina, which is the interesting one. And what I'm going to try to do otherwise is a bit of an overview, which maybe pulls together themes from the meeting. But I tried to predict in advance what the themes would be. So they may not correspond very well with the talks. And they're certainly not in the order in which things appeared in the talks. What I've been trying to do during the meeting is to slot in references to species systems that have been described during the talks. So I think they're almost all in there somewhere now, apart from Alex's. Sorry, Alex. But don't be offended if I don't refer to your particular subject. I've put ecological in brackets here because personally I don't like categorizing speciation into ecological speciation or gene order speciation or allopatric speciation or hybrid speciation or whatever, because I think in most speciation events a variety of different processes are involved. So what we've been talking about here are often cases where divergent selection in different habitats is a key component of speciation, but it's far from being the only process involved in the completion of speciation in those examples which start with local adaptation. So yes, there may be strong ecological drivers in some cases and relatively weak ecological components in others. I think it's generally more helpful to think about the contributions of those different components than to ask whether this particular case is sympatric or ecological or whatever badge you want to put on it. So that's my excuse for putting ecological in speciation. Ecological had to be there because it's the title of the meeting. Well, sort of divided up. And I hope you'll see that the sort of various topics that come up in this talk are relevant to all forms of speciation, not just to ecological speciation. So the other thing I wanted to say by way of preamble is that a couple of weeks ago, some of us here were at a Prospex workshop on genetics and genomics of speciation. And I've stolen ideas from the discussions at that meeting. And so those who were there will sort of see that some of the themes that I've structured around derive from that meeting. And hopefully you'll all be able to share in that meeting because we're trying to write a paper from it, which may or may not appear at some stage in the future. It's a 30-author paper, so it's not quite clear how it's going to turn out at the moment. So I'm going to start by stealing an idea, actually partly from Paul Hohenlohe, who was at that meeting, and asking you to contribute to a small game. So what do you see in this picture? Shout out if you see something. Sorry?
An insect.
An insect? Any advance? Pelvic girdle, yeah. An aphid, yeah. Yeah, okay. Lots of possibilities. It's actually just an ink blot, and some people might recognize it as coming from the Rorschach ink blot test. And the suggestions you make as to what you see in the picture are supposed to reflect your personality. I'm not sure what that says about Dolph and pelvic girdles, but there you go. It's an example of apophenia, which is the sort of ability of humans to see patterns in what are actually random arrangements of things, and that's relevant to this sort of issue. So what do you see in this picture? The pelvic girdle, yeah. Evidence for selection, and what Paul asked us to do at this workshop was to say how many genes are under selection in this picture. How many genes are under selection? No, it's hard to say, isn't it? Some might say 1 because of this peak here, and some people might say 1, 2, 3, 4, 5, 6, 7. I don't know. It's hard to interpret these pictures. One of the other things about apophenia is that it's quite easy to change people's perception of the otherwise random patterns. So this is an example. Putting the banana here helps us all to see the monkey. This is actually a monkey god which receives lots of tributes. at the moment. But really, of course, it's just where a car happened to damage a tree. So I can change your perception of that other picture simply by drawing my line somewhere else. Now there's definitely only one gene under selection, isn't there?
There might be 20 genes in that field.
Yeah, I mean, the point is that it's hard to interpret these genome scan patterns. I was actually expecting to see more of them here than we have seen, but I'm pleased to say that we have seen a few of these squiggly lines, as Sebastian called them. Yeah. And the problem we have with these patterns of genomic differentiation is that what we're interested in is the barriers to gene flow. So we're trying to use these to detect barriers to gene flow, but there are actually many other processes that underlie the pattern of differentiation between any two populations that we may care to investigate. So obviously the demographic history has an impact on how much variation in FST we see, as well as the overall level of FST. The pattern of recombination influences the pattern that we see, whether we plot this by genetic distance or physical distance. The amount of the importance of background selection has an impact. Background selection can actually emphasize FST by reducing within population variability, for example. The hybridization can have an important impact by making some populations more different from others because they contain genes by hybridization, for example, and also in other ways. And the extent of standing variation before selection started also influences the signature of selection that we see after selection that has operated. So there are lots of things which are essentially nuisance parameters, if you like, which we have to get rid of before we can get to the thing that we're actually, there was one more, I forgot about that one, there's mutation as well. Variation in mutation rate, again, as Sebastian mentioned, can influence this pattern. These are all nuisance parameters. What we really want to know is the pattern of barriers to gene exchange along here. And it's really difficult to extract that signal from the pattern of differentiation. So that was really the sort of first major question area that I was sort of identifying, that we've been discussing, and it's come up in various talks during the meeting. How can we extract as much information as possible from patterns of genomic differentiation in order to understand barriers to gene flow that exist at different stages in the speciation process? And I just put some thoughts down here, some of which, as I say, have come up during the meeting. But one possibility, of course, is simply to refine our description. Okay, so not just look at gene flow, but to look at other signatures, other measures of differentiation, or to look separately at differentiation between population and variation within populations, for example, to get a better description of the pattern than we see just by looking at FST. And Felicity gave us a nice example of that with the haplotype blocks in sticklebacks. By looking at that level, we can see more than we can if we just look at the FST patterns. And I think one of the things we need to think about is more ways of doing that sort of thing. We can also try more refined analysis. So, for example, we can make relatively parameter-rich models which try and separate out some of those various nuisance effects so that we can get at the ones we're really interested in. The problem with having these sorts of models is that you need more data if you're going to fit more complex models. So we need to be able, if we're going to factor out these other possible effects, we need to know much more about the populations we're working with than just the pattern of genetic differentiation between them. And so we've seen progress in that sort of direction in various talks. So recombination rate variation is obviously an important factor, and we've seen both in sticklebacks and in sunflowers that the pattern of recombination across the genome helps us to understand patterns of differentiation. And we also saw we're reminded in the talk about dogs that recombination rates evolve, so they're not a sort of fixed feature of the genome. And during speciation, we may well expect recombination rates to change, and that's something we have to bear in mind. We might want to know about gene density and the extent of constraint on genes because that's what determines background selection. And on the whole, that's rarely been incorporated into these sorts of analyses so far, but it is important. Mutation rate variation we saw in sunflowers can influence the pattern. It's actually a relatively easy one to get at least to a first approximation, because you can look at outgroups and get some sort of estimation of mutation rate variation across the genome. We should also not forget that the demographic history influences the pattern of differentiation, and that there may be, in many cases, lots of external information about demographic history that ideally we would try and use in our modelling process along with the genetic evidence, so the age of our islands and the archaeological evidence about when mice were in one place and not in another, for example. And we were also, I should have put aphids in here, reminded that actually in many cases, although we tend to make pairwise comparisons because they're easy, populations don't actually, in the real world, exist usually in pairs. So Pierre in his talked about multiple races of aphids. Now that's a complication that most of us try and avoid, but it is actually the real situation that there are more than just two populations exchanging genes. Another thing we can do, of course, is have better theory, and Sebastian again referred to improvements on the theory behind divergence hitchhiking, for example, which suggests that it may be an important factor only in a limited number of cases, but various types of improvement in theory will help us to understand these patterns. Perhaps most powerfully, we can rely not entirely on these patterns of differentiation, but combine them with other sources of information. And again, we've seen good examples of that sort of thing. We can combine scan approaches with knowledge about candidate gene groups, and I'll say a bit about that in aphids, but we've already seen a bit from Pierre. We can combine QTL mapping with, or admixture mapping with our genome scans, as we've seen in sticklebacks and in senecio, for example. We can combine with functional analysis, as we've seen in sticklebacks and in cichlids. Sticklebacks I've had to put in rather a lot of places, I'm afraid. Ideally, we can also use experimental manipulation, so to look more directly at selection, rather than simply inferring selection indirectly by looking at the patterns it generates. Ideally, we should experimentally manipulate the environment or the populations to look at the effects of selection. I'll mention an example of that in a moment. So this is one of the cases where I'm going to digress a bit into the boring species. In P. aphids, as Pierre suggested, host acceptance is a key determinant of adaptation and assortative mating, and it's important that it contributes strongly to both of those. Acceptance of the host plant determines the environment in which the aphid has to survive and grow and produce offspring, and it also determines who they mate with, because they stay on the same host in the case of P. aphid for mating. And we know that host association in aphids is mainly determined by chemosensory cues that the aphids receive after landing on the host plant. And that immediately identifies chemosensory receptor genes, or genes involved in the chemosensory reception, as a strong group of candidates. Unfortunately, those families of genes have now been annotated in multiple genomes, both insect and vertebrate genomes. And so they provide a good set of candidates. But they are large families, and so one still needs approaches to identify within that large candidate family the subset of genes that are likely to be directly involved in host acceptance differences between races. So P. aphid has about 150 genes in total in the odorant receptor, gustatory receptor, and odorant binding and chemosensory protein families. But it's possible to study all of that set of genes now by using capture techniques. So we know the genomes, the sequences from the genome. We can capture that set of genes and sequence them en masse along with control genes, and that's what Carol Smadger and I and others have done for 3 races, and Pierre referred to these data. So starting with that subset, that set of 150 candidate genes, we can use the sort of FST, standard FST outlier approach to narrow the set of candidates down to a relatively small group of loci, which actually turn out to be predominantly odorant receptors rather than gustatory receptors. There are a few gustatory receptors in there, but none of the odorant binding or chemosensory proteins, which are at an earlier stage in the reception process, were identified as outliers. And actually only one of the control genes in this set was identified as an outlier. That's the RAD51C that appears there. Actually, going from here to understanding the function of individual chemosensory genes is actually quite difficult, because in general the ligands of these genes are not known, and so it's quite hard to associate these functionally with the differences between the plants. That's something we're actively working on. The other thing I should say is that, as Pierre mentioned again, although these are strong candidates, they're probably not the only genes involved. And one of the problems with candidate approaches is that they focus your attention possibly too much on a set of genes which you think a priori are important. So Pierre mentioned this scan using microsatellites, which found a subset of outliers, some of which he's now found to be common to differentiation between other races. Only 2 of these 11 outliers were chemosensory genes. So there are clearly other things going on. And actually the 2 that were chemosensory genes don't correspond to any in the previous diagram, which is a slight embarrassment to us. But anyway, so to continue this digression, we're currently expanding the capture sequencing based outlier analysis of candidate chemosensory genes to more races and larger samples of individuals. So this is just to say what data we have available. But we don't have an answer yet in terms of whether the same chemosensory genes are involved in this larger sample of races. And part of the reason for that is because we've discovered an interesting, although slightly irritating, feature of the data. And that is that many of the genes involved show copy number variation. We initially detected this because many of the SNPs didn't show 50-50 segregation, 50-50 ratios in heterozygotes. But if we look at analyze the coverage of different genes and compare that between races, we can see evidence for copy number variation. So some genes behave nicely. If they're behaving nicely, they'll all be in this pink band here, which suggests that there's one copy everywhere. Some of them, you get a few individuals like this, which show apparently 1.5 copies, so perhaps heterozygous for a duplication, whereas most genes are behaving normally. Some genes are just rubbish, and presumably there's something wrong there. Fortunately, that's not a very large category. Some genes show evidence for duplication or loss in just some exons rather than in the whole gene. But most interestingly, from our point of view, some genes show this pattern where the red race has more copies than the majority of races, and the black race has fewer copies. Actually, this is all reference to Medicago, which is the race for which the reference genome sequence came. And the fact that these are 1/2, 1, and 1.5 actually suggests that the gene is duplicated in Medicago and has been misannotated as a single heterozygous locus. So probably this is 1 copy in this race, 2 in all of these, and 3 in this race. So it's early days to say whether this is important in differentiation between races. But I think it emphasizes that one of the other complications we have to worry about is that it's not just SNP variation that differs within and between species. We need to keep our minds open to the possibility of copy number variation, insertions and deletions, and of course rearrangements. I said I would say something about experimental approaches, which hopefully, you know, are a potential route to giving us better insight into which of our outlying loci are really outlying because they're under selection. That was the logic behind this experiment, which was conducted by Patrick Nosal and Jeff Feder and their groups, which is currently submitted. Patrick said it was okay to tell people about it. What they did was to move Timema stick insects from a variable population onto one or other of the host plants, and they did that in various replicates, including at different altitudes. And then they did that with young individuals, and they genotyped those individuals by taking a small sample from the leg, and then they regenotyped the ones that they captured at the end of the generation, so they can see what gene frequency changes there have been actually within generations. So it's only a part of the total selection that they're able to look at. And then they genotyped everything with rads. And I think what they were hoping to find was some subset of the outliers that you see in a genome scan, which were nicely under selection. Actually, what they saw was a huge number of loci that changed in frequency more than you would expect by drift or by chance sampling process between the release and the recapture. So all of these loci up here are more different between the pre-release and post-release. So in a sense, this, well, maybe it hasn't helped, or maybe it said that actually there is selection operating on a huge number of different regions of the genome. And one of the striking features of this was also that much of this selection doesn't seem to be related to the difference between host plants, but to other differences between environments which they weren't expecting. So that's also something that we need to bear in mind, is that often we focus on a particular environmental axis because it's the one that strikes us initially, but there are often many other environmental axes that we're not thinking about and which may be contributing to selection. Okay, so to introduce the next sort of set of questions, I want to move to the interesting organism, Littorina saxatilis, although some people here would say Littorina fabalis is the interesting organism, I guess. Yeah. So Littorina saxatilis, as many of you will know, has so-called wave-adapted and crab-adapted morphs on many rocky shores around Europe. The wave-adapted morph has a relatively large aperture, accommodating a relatively large foot, which helps it to hold on. But it has a thin shell and is relatively small. The crab-adapted one is relatively large, has a robust shell and a relatively small aperture, which makes it more difficult for crabs to get their pincers in. And saxatilis is ovoviviparous. It retains its embryos, and they crawl away as little babies, so it has relatively low dispersal. And a particularly interesting feature is that we see a pair of morphs that are relatively similar in at least 3 different areas of Europe where they've been studied in detail, the UK, Spain and Sweden, where the details of the habitat difference vary in that in the UK the boulder field is in the mid-shore here where the crabs are, and then the upper shore has the strong wave action. In Spain the strongest wave action is low on the shore, so the wave type occurs low on the shore, and the crabs occur further up, so that crab type occurs above. And in Sweden there's effectively no tide, just about 30 centimeters tide in this part of Sweden. So the differentiation is along the shore. You get boulder-strewn bays and rocky headlands. So this is a nice example of parallelism, and I'll come to the parallelism bit in a minute. Actually, I'm getting ahead of myself there. What I wanted to do first was to look at the types of barriers to gene flow in this system. We have to speed up a bit, actually. So the things that operate against gene flow between morphs in Litrinus saxatilis. So first of all, things that affect local adaptation, and then phenotypes that affect assortative mating, and then potentially intrinsic incompatibility. So we have evidence for all of these, but there's still some debate about whether there's any intrinsic incompatibility in this system. And what's important for progress towards speciation is that these barriers work together, that they're coupled. Yeah. It's actually easy to understand how these things work together, because selection across the same transition works on all of these traits and therefore holds them together, maintains association between them. So coupling of barriers caused by different phenotypes that are adapting to the same pair of environments will sort of occur automatically. Coupling of mate choice with size is partly caused by the with local adaptation is partly caused by the fact that mate choice operates largely on size. So size is what we would call a multiple effect trait. It contributes both to local adaptation and to assortative mating. But of course, there also has to be some size preference, which has to be maintained also in association with the phenotypic differences, the other phenotypic differences. It's possible that some of the association is not necessary because the traits involved are of the one allele type. So we don't know this, but habitat choice could be determined by some behavioral rule, which is the same in both environments, like stay where you have high fitness or something like that. But it could also be a two allele type system, in which case something else would have to maintain coupling between the selected traits and the habitat choice. Then, of course, it's possible that associations between these various things are built up by reinforcement, so would require linkage disequilibrium. And then reinforcement is part of a sort of more general coupling process. So coupling could be maintained between intrinsic incompatibility and these extrinsic forces of selection, simply by the fact that different barriers tend to attract one another. So the point of this digression is to say that if we're interested in speciation, we're interested in much more than just those traits which are under divergent ecological selection, because many other things contribute to barriers to gene flow. Some of those other things may be under divergent selection, but many of them may be under indirect selection, as through reinforcement, for example, or there may be no selection for differentiation, as in the example of one allele traits. Or they may have evolved for completely other reasons and just be trapped at the barrier which is created by the ecological differentiation. So if we're looking for barriers to gene flow, we have to be able to detect these sorts of contributions to the barrier as well as those that respond just to divergent selection. So I think a really important set of questions relate to what are the phenotypes that contribute to reproductive isolation, and of course in what order do they evolve. I will try and speed up a bit. So barriers may be intrinsic or extrinsic or assortative, and we've seen examples of those various different types of barriers here, although we've concentrated on the extrinsic ones. We need to remember that the other ones are out there, particularly these assortative ones, habitat choice and mate choice and so on. And we need to know what different signatures to look for from these different types of barrier. We need, in particular, to look for evidence of coupling of different components of isolation. And I think this is a shortcoming of the basic genome scan approach, that it doesn't look for these associations between different things that cause isolation. And we've been reminded a couple of times during the meeting about the importance of sexual selection, which may produce divergent selection, signatures of divergent selection which aren't closely linked to the environment, and may cause more rapid divergence than environmental-based selection. And coupling we will often see through patterns of linkage disequilibrium or epistasis, And so we need improved ways of looking for those things, and both in sticklebacks and in Petrie's new LD methods, we see that there is progress towards finding associations between traits that are under selection or genes that are under selection, rather than just looking at them individually. And of course, we've been reminded various times about the importance of suppressed recombination in maintaining these associations, and that includes the importance of sex chromosomes in speciation, which was mentioned in the nice moss talk. Monoallele traits don't leave obvious genomic signatures, and so we need to be alert to the fact that they may be important in speciation and are not easy to detect. And we've also been reminded a couple of times about the importance of plasticity, but perhaps in promoting speciation, perhaps in impeding it in some cases. To find out the order in which different reproductive barriers evolve is a very difficult thing to do. We need a strong historical framework in terms of understanding the history of the populations and maybe inferring it from genetic data. It would be nice to know the ages of alleles, as was discussed in the stickleback case again, and it would be nice to use comparative approaches across the speciation continuum. And therefore, in that case, systems like the aphids, where there are different degrees of reproductive isolation within the same system, are particularly powerful. Just to go to parallel divergence, which I mentioned in the case of Littorina, we can show that the morphological differentiation in the three regions of Europe is actually very similar. So in all three cases, the populations, which way round is this? The wave and crab populations differ in size and on the same shape axis. They differ more in Sweden than in the UK, and sort of slightly intermediate in Spain. But it's clear that phenotypically there's parallel response to the different environments in Littorina. But what's less clear in a case like Littorina is whether these populations originated demographically in parallel, or whether they originated once and then spread to the different regions, to take two extreme possibilities. It's not like a case where we have extremely isolated islands or lakes where repeated colonization is perhaps very unlikely. In general, in cases where we see parallel divergence, I think it's really important that we establish clearly whether they particular localities were colonized by one population and then diverged into two ecotypes, or whether the ecotypes diverged and then colonized the populations separately. We need to answer that demographic question before we start asking about the genes that are involved and whether they evolved in parallel or not. So in the Littorina case, we've been trying to do that more formally than is usually done by comparing essentially these two historical models, both of which are rather complicated. I'll take you through the basis of them. Basically, the parallel divergence model says that each locality was colonized by some population, and then within the locality, the two populations of the two morphs split from one another. The alternative, which we call the old divergence model, the two morphs, the two ecotypes separated at some point in history somewhere, and then they colonized the two different sites we're comparing, which may be either two sites within a region or two regions of Europe. They colonized those sites separately. And in this model, we allow the possibility for there being some period of allopatry around the origin of the original morphs, and then there's migration going on all of the time. So this isn't really an allopatric sympatric comparison. It's an old divergence versus parallel divergence comparison. I think it's important to try and establish which of these two models is a better fit. And we've tried to do that with old style data. So we've actually used a combination of mitochondrial DNA sequence, single 3 nuclear sequences, and AFLPs to compare these two models. And actually the parallel scenario is supported over the old divergence model quite strongly, and that's true both for comparing pairs of localities within regions and comparing localities in different regions. So it looks like the sites were colonized essentially by one population, which then subsequently divided into two separate ecotype populations. That doesn't tell us about the genetic basis of the differentiation. It simply tells us about the demographic history. To look at the genetic basis of the population, we've been doing a sort of poor man's survey of the extent of parallelism. So this analysis I'm going to show you, which is still sort of ongoing, is based on RNA-seq from two pools of individuals from each of the two ecotypes in each of the three regions, so 12 samples in total. And it's been analyzed at the moment using, by mapping those RNA-seq reads to the embryonic Littorina genome assembly, which actually consists of there are about 70,000 contigs, which essentially correspond to exons or, in some cases, whole genes. And we've mapped the transcriptome reads to those, and then analyzed them using population, the Schlotterer package, to give us an FST value per contig for about 7,000 contigs. And because we had two replicates from each ecotype in each site, we can check for the noise that's introduced by that analytical process by looking at the FST distribution between replicates, which is this distribution here, compared with the FST distribution between ecotypes in the same population. And so replicates are always very similar and don't have these highly divergent loci. And but the question we're interested in here is, if we look for outlying contigs, so contigs that are more differentiated between ecotypes, or most differentiated between ecotypes, are they shared between ecotype pairs in different regions? One way we can do that is simply to use quantiles, as Sebastian did in his case. This is for the 98% quantile for the UK-Sweden comparison. And you can see that there's a set of outliers in Sweden, or outliers. There's a tail of values above the 98th quantile in Sweden and a tail of values in the UK. And there's a relatively small number of contigs which are outliers in both cases. It's actually more of these than you expect by chance. But it's quite hard to tell from a plot like this what that says about the extent of parallelism. So we need some reference against which to compare that. And we need to be sure that the pattern doesn't change as we change this cutoff. So we've used various cutoffs in rather the same way as Sebastian did. And we've compared it with two sort of reference points. So this plot needs a bit of talking through. So on this axis, actually convergently with Sebastian, is the ratio of observed to expected shared outliers. And these points through here are the three possible comparisons between the three regions: Sweden, UK, Sweden, Spain, and so on. So there are more than expected, which would be one to one here, but not very many more than expected, and it's rather similar for each of the three country comparisons. And then this upper line, these upper lines are when we take the two replicate pairs from any one site, and there we expect all the real outliers to be shared because they're just replicate pairs of populations. So if we had 100% sharing of parallel evolution, if you like, we would expect these points to be up here. And then these lines down the bottom are doing the sort of orthogonal comparison where we take, we compare the two samples, which should only have outliers which are noise, and we compare those between ecotypes where there should be no sharing. So this is the sort of null expectation, if you like. So in a sense, the position here is a measure of the proportion of shared outliers. Yeah, and there are some, but it's quite a small proportion, which is actually, I think, rather similar to Sebastian's result. So I'm running out of time, so I'm going to have to speed up a bit. So there's obviously an interesting set of questions about whether repeated evolution is commonly parallel, convergent, or concerted. And we've seen, first of all, you need to establish the demographic history. There may be parallel reuse of alleles. There's some evidence for that in sunflowers. Or there may be what we call concerted evolution, where the same alleles are reused, having been passed between different cases of divergence. Or there may be convergent cases where different alleles are used, either at the same or at different loci. There's a whole series of possibilities that we need to distinguish. And we're getting hints about various of those processes occurring, but we have very little idea about the generality at the moment. More generally, I think if we take the focus away from individual loci, we need to know more about pleiotropy and constraints and about the importance of genetic correlations between traits. That's been emphasized in various talks. And then rather briefly, just to finish off, I want to say that there's also a set of questions about the speciation continuum, how speciation gets started and completed. I think we need to know more about the very early stages. The cases that we think of as early stages of speciation actually tend to have really quite high reproductive isolation. And I think there may be, and they're also a very strongly selected set of comparisons, because we pick on things where we see differentiation, very obvious differentiation between pairs of habitats, and we focus on those. So we don't know much about the sort of generality of divergence at the early stages of separation of populations. There's an interesting, I only say this because I'm an author, paper surveying outbreeding depression studies. And outbreeding depression is actually, if you viewed from our perspective, the very beginning of speciation. And actually one of the main messages of this is that there's not very much data out there to tell us about it, but outbreeding depression is common between recently separated populations, even when they're not obviously in different environments. So I think speciation starts at that point. And we also need to know about the very late stages. This is a very difficult bit of speciation to study. Once gene flow is reduced to 1% or 0.5% or something, how does it eventually get finished off? And we also need to know about the resilience of species. Are there things which alter the potential of new pairs of species to persist and contribute to diversity? And we've had some very interesting talks about what determines patterns of diversity and what the correlates are of diversity in different parts of the world and in different taxonomic groups. One interesting question that we've sort of tried to pose in the past is whether speciation mechanisms or the genetic basis of speciation relates to the patterns of diversity that we see. And I think although there are some fascinating things coming out of the diversity or diversification rate analyses, they don't necessarily get directly at this question of whether there's any relationship with speciation mechanism, although there are some hints, I think. So in the back talk, it was emphasized that coevolutionary pressures might be important in diversification, and it's also possible that the sort of open-ended nature of coevolutionary changes might be important in the speciation process itself. So I think in the interest of time, I'll skip these two examples that I was going to talk about briefly. Just go on to say that this set of questions that I've discussed is by no means exhaustive, and I hope that you're all now thinking, he didn't say X, Y, or Z, and that in the forthcoming roundtable discussion, you'll present lots of questions that I've failed to include here, or answers to some of the questions will be even better, of course. Before I stop, I want to say thanks, first of all, as others have done, to Godfrey, without whom many of us wouldn't be here and wouldn't be thinking quite the way we do. And then, very particularly to the organisers of this excellent meeting. It's been a great couple of days. I've heard lots of people saying so in the coffee room quite without prompting. It's been a lot of fun, so thank you. And then to the participants of the Prospects workshop, whose ideas I've stolen, to Litorina people, to aphid people, and to those who've given talks and posters, and the rest of you for your attention. So thank you. Yeah, at some stage in the future. It's a 30-author paper, so it's not quite clear how it's going to turn out at the moment. So I'm going to start by stealing an idea, actually partly from Paul Hohenlohe, who was at that meeting, and asking you to contribute to a small game. So what do you see in this picture? Shout out if you see something. Sorry?
An insect?
An insect? Any advance? Pelvic girdle, yeah. An aphid, yeah. Yeah, okay. Lots of possibilities. It's actually just an ink blot, and some people might recognize it as coming from the Rorschach ink blot test. And the suggestions you make as to what you see in the picture are supposed to reflect your personality. I'm not sure what that says about. Hi, thank you. Actually, I'm just going to have a couple of asides about projects in my group. One about aphids, which is the boring one, and one about Litorina, which is the interesting one. And what I'm going to try to do otherwise is a bit of an overview, which maybe pulls together themes from the meeting. But I tried to predict in advance what the themes would be, so they may not correspond very well with the talks. And they're certainly not in the order in which things appeared in the talks. What I've been trying to do during the meeting is to slot in references to species systems that have been described during the talks. So I think they're almost all in there somewhere now, apart from Alex's. Sorry, Alex. But don't be offended if I don't refer to your particular subject. Particular case is sympatric or ecological or whatever badge you want to put on it. So that's my excuse for putting ecological in speciation. Ecological had to be there because it's the title of the meeting. Well, sort of divided up. And I hope you'll see that the sort of various topics that come up in this talk are relevant to all forms of speciation, not just to ecological speciation. So the other thing I wanted to say by way of preamble is that a couple of weeks ago, some of us here were at a Prospects workshop on genetics and genomics of speciation. And I've stolen ideas from the discussions at that meeting. And so those who were there will sort of see that some of the themes that I've structured around derive from that meeting. And hopefully you'll all be able to share in that meeting, because we're trying to write a paper from it, which may or may not appear dull from pelvic girdles, but there you go. It's an example of apophenia, which is the sort of ability of humans to see patterns in what are actually random arrangements of things, and that's relevant to this sort of issue. So what do you see in this picture? The pelvic girdle, yeah. Evidence for selection, and what Paul asked us to do at this workshop was to say how many genes are under selection in this picture. How many genes are under selection? No, it's hard to say, isn't it? Some might say 1 because of this peak here, and some people might say 1, 2, 3, 4, 5, 6. I put ecological in brackets here because personally I don't like categorizing speciation into ecological speciation or gene order speciation or allopatric speciation or hybrid speciation or whatever, because I think in most speciation events a variety of different processes are involved. So what we've been talking about here are often cases where divergent selection in different habitats is a key component of speciation, but it's far from being the only process involved in the completion of speciation in those examples which start with local adaptation. So yes, there may be strong ecological drivers in some cases and relatively weak ecological components in others. I think it's generally more helpful to think about the contributions of those different components than to ask whether this particular